All right, ladies and gentlemen, so welcome back. We are going to continue our discussion on the applications of biotechnology by talking about cloning today. So, a couple of things about the way that we talk about cloning. First off, cloning is just copying of cells. Okay. So when we talk about making a clone, we're really talking about making copies of a cell rather than truly making a copy of an entire organism. And as we continue talking, hopefully that'll make a little more sense. So I'm going to talk you through in just a second, really about the process of the way this works using Dolly the sheep, which was the first successfully cloned organism um, in the UK. So the basics when we talk about cloning cells, well, I've got to take DNA out of an adult cell. I've got to take that DNA, essentially the nucleus, insert it into a embryo that I've removed the nucleus from, and then I have to implant that embryo into the mother that then gives birth to the organism that is genetically identical to the original adult that I took the DNA from. Here's the thing. While we are getting better at cloning, there are still issues with talking about being able to successfully clone an entire organism. We're getting pretty good at doing it in tissue samples, so just chunks of cells. But being able to successfully clone an organism is still challenging. And the reason really is because those genes, when we talk about development of a new embryo, there are a set of genes that need to be turned on and turned off during development, particularly um, in sperm and eggs, in the way that they develop in the adults before they get fertilized, as well as in the embryo once the fertilization process happens, and then even in you as you continue to grow and develop from baby to adult. All of those are very tightly coordinated, these sort of on off switches that need to get turned on and turned off at the right times. And the problem that we're finding with cloning is that it's not easy to necessarily replicate when I'm taking genetic information out of an adult and trying to put it into a brand new organism as a baby. We're sort of missing that step of coordination of genetic information pre fertilization because those switches have already been turned on or turned off in that adult. Hopefully that makes sense. So as a result, we have a really limited number of clones that have successfully survived for long periods of time. And Dolly the sheep is definitely a good example of sort of, we were successful-ish because she only ended up living a few years before she ended up dying, really, of a lot of complications that we would typically associate with older adult sheep. But if we take a look, here's the process of how cloning works, right? So I'm going to take my sheep, right? Some reason I want to clone it. It's a really good livestock organism. I want to make lots and lots of copies of it because it really has been sort of the pinnacle of my sheep, right? So we'll take some cells from that sheep. In the case of creating Dolly, we did it from their little udder, took some samples, took it back into a lab, and here's that donor cell. We're going to extract that donor's nucleus, right? So I'm going to take that nucleus out, take that DNA out, and now I've got the DNA from this original donor. I'm also going to go to another female sheep and extract egg cells. So here's a bunch of egg cells that I'll extract, and then I'm going to suck the nucleus out of them. So we basically create what they call blank egg cells, which is what you're looking at right here. So here's my donor nucleus plus that blank egg cell. Now, in a petri dish, if I do this correctly with a little electric shock, I can force that nucleus into this blank unnucleated cell, right? So blank cell, no nucleus, nucleus from my donor. I now have a embryo that can start, or I should say I have essentially what would be a fertilized egg, right? I now have all the genetic information necessary to make an adult in this cell here. Allow that cell to start dividing in a petri dish, create an embryo, so a bundle of cells that then can be placed in the uterus of the mother That mother then will grow up that sheep, and here's your cloned lamb. So this lamb is essentially genetically identical to this one. Okay. That being said, keep in mind we're talking about genetically identical in the sense that they have the same A's, T's, C's, and G's. But there are plenty of things that we know, go back to our discussion about epigenetics, and recognize that there are a lot of environmental impacts and factors that do inform the way our cells express genetic information. So it's more above and beyond just the sequence of nucleotides that you inherit, but it's also environmental conditions that dictate the way your body uses that information. So when we go ahead and create that clone, that lamb is genetically identical to that donor, that adult that we got that genetic information from, but it doesn't necessarily mean it's going to be identical. And think of it this way. Let's just say we were going to go and make a clone of you. Well, we clone you, great. So we're going to take genetic information out of your skin cells, for example, and place that genetic information into a blank cell. But now that blank cell has to become an embryo that gets implanted into a mother, and 9 months later, we get a baby you. So think about all of the experiences that you have had in your life. There is no way to replicate that for now baby you. And so that new clone will look like you, will sound like you, will very likely have certain things in the way that they function that are like you, but there's really no true way to make an exact copy of you. And the other misnomer when we talk about cloning is that kids assume, well, it's going to be 15, 16-year-old me when we make that clone. Well, that's not true. It's going to take 15 years for that baby to now become 15-year-old clone you, and you are now 15 years ahead of that. So just some interesting things to think about. So definitely technology that's getting better. It's a place to look, especially when we talk about tissue samples and being able to maybe make um, organs for transplant issues, or even the livestock, right? If there are certain traits that we're after, this may be a viable option. But unlike maybe the movies and Hollywood's version of cloning, we're not going to just create another you.